Hello, everybody. Welcome. Good afternoon. So we'd like to get started with the kickoff for this year's Timothy Johnson Medical Scholar Series. It's my pleasure to introduce the series, and then I'll introduce Dr. Vaughn, who will introduce our speaker. So it's a double intro today. Just by way of a quick background for those of you who don't know or don't remember, this particular series is named after Dr. Timothy Johnson. Dr. Johnson was one of the founding faculty members here at the Virginia Tech Carilion School of Medicine and was instrumental working with Dr. Berry and others in getting the curricula, the programs going. And Dr. Johnson, who is a basic scientist himself and a very good one, had a real passion and commitment to learning about breakthroughs and discoveries and how science led to medicine and how well integrated they were. And he sadly passed a couple years ago and was here, thank goodness, for an event where we kicked off the series in his honor and for his name. And so we're extremely honored to remember him today and all he did to get the medical school going here. And this series is a living memorial for all that he did for this medical school. So it's a real honor to be part of that and share that with all of you. And just by way of reminder, the next talk in the series will be September 28th. Dr. Nadler will be here, Jerry Nadler from Eastern Virginia Medical School. So I'm going to introduce Dr. Vaughn to introduce Dr. Pace, but first I just want to say one thing about Dr. Pace. Hope I don't ruin your introduction. So Dr. Pace, for all her recognition and accolades and achievements, and I imagine you'll hear some about from Dr. Vaughn, one that I'm very close to is that she has been recognized by a society called the Society for Experimental Biology and Medicine that she's been very involved with. And I served as president of that society for a few years, not too many years ago. And one of the awards she got from that society was recognition called the Bert Sobel Best Presentation Award. And Dr. Sobel was one of the giants in experimental biology and medicine and a great leader in that area. And as the president of that society, I know what was involved with selecting people for that recognition, and so that's a great recognition for her, amongst many others that she has. And I'm going to make one other quick announcement today. How many of you know what the Society for Experimental Biology and Medicine is? Don't be shy if you don't. Okay. For all of you students out there, any of you who want to join today, I will pay your membership for a year. For a year, I will pay your membership. I'm writing a check today for the total amount. And if you just give Dr. LeConte at the end of this session your names, if you want to join, it's one of the oldest professional societies that brings together biology and experimental medicine. It has a great legacy, and they have great meetings and have a great journal and a lot of other good things. And I think it sort of represents the confluence of science and medicine that we try to embrace here very much. And of course, Dr. Pace represents that very much so. So check with Dr. LeConte after, and now I'll introduce Dr. Jen Vaughn, who will introduce our speaker.
Thank you, Dr. Friedlander. It is my absolute honor to welcome Dr. Betty Pace to the Virginia Tech Carilion School of Medicine. She's visiting us from Augusta University in Georgia, where she currently holds the title of the Francis J. Tedesco Distinguished Chair of Pediatric Hematology and Oncology. Dr. Pace is a native of Wisconsin, where she told us last night she grew up as number 10, was it, out of 15 siblings, so maybe one of her greatest achievements among many. She completed her Bachelor's of Science degree at Marquette University in Milwaukee, followed by obtaining an MD at the Medical College of Wisconsin, where she also completed her pediatrics training at the Children's Hospital there. She then went on to complete a subspecialty fellowship in hematology and oncology at the University of Colorado in Denver. And I'm assuming that this is probably where she developed her love of research, so went on to do a postdoctoral research fellowship at the University of Washington soon thereafter. After completing her training, she has forged a very impressive career for herself, holding academic positions at MCW Children's. And I can just say this institution finds her to be so beloved, they've endowed a scholarship in her name there. She has held positions at the University of Southern Alabama and at Texas in Dallas. And as I mentioned before, she now holds full professorships in both the departments of Pediatrics and Biochemistry and Molecular Biology at Augusta, where she dedicates her clinical time to running a sickle cell clinic and also managing a telemedicine clinic that serves sickle cell patients in rural Georgia. Her research, as many of you have learned about in preparation for this talk today, has focused on elucidating mechanisms of globin gene regulation and has led to multiple publications in high-impact journals. And she's also been the recipient of a substantial amount of funding from the National Heart, Lung, and Blood Institute. She's been honored with multiple awards, including the one that Dr. Friedlander mentioned. It was difficult for me to choose which ones to highlight, but I will say she was given a Lifetime Achievement Award for contributions to science from the Sickle Disease Foundation of California, and came to my attention when she was recently honored at the American Society of Hematology, receiving the first ever award for leadership and promoting diversity. And just a quote from the ASH website as to why that award was established. It is to recognize hematologists who have supported the development of an inclusive hematology workforce, encourage the career development of underrepresented minority trainees, or made a commitment to inclusiveness through contributions in support of ASH's mission. So I could go on, but then she wouldn't get a chance to talk. So I'm going to welcome Dr. Betty Pace now.
Thank you both for such a kind introduction. And it sounds like I have a lot to live up to now, this introduction, but I'm really honored to be here today. And I didn't know this was the first lecture of the year before I came, but I'm hoping that I'll be able to share with you some insights and things that I've learned along the way, some of our research accomplishments, and then end up talking about some of the work that I've done with training and why I earned that ASH award. For disclosures, I have done some work with Bioveritin, was previously known as Biogen, so I'll show some data just as full disclosure. And today I wanted to basically kind of take you through my story. The three important components: personal, family, influence, education, and professional training. And then my physician scientist career path and some of the decision making and why I chose to go and make certain decisions along the way that may help you when you get to those key points in your training to help you make the right decision that's going to affect you, basically for the rest of your life. One word about family and friends. I could not have done this without my family. Yes, it is true. I'm number 10 out of 15 children. So we had a large family. You had to be pretty strong and aggressive to make it. But I was the only one who chose to go into medicine. But I've been fully supported for years by my family. My father had an incredible work ethic that he passed on to me, which is required to be successful in medicine or in research. So I really appreciate my family and the influence that they had on my career. And then also, I was asked a question at lunchtime, what made me choose sickle cell disease research? As a teenager, I had a really good friend who died from sickle cell disease. So this moment, deciding moment before sickle cell disease had good treatments, really influenced and helped me make my decision to follow my career path, to do research related to sickle cell disease, and later as a physician scientist. We've gone over a little bit my different training points over the years, but I wanted to share with you a little bit about my decision making during those different points. I went into college with the intent to become a biochemist, to get a PhD in biochemistry. And it was the influence of my now mentor and very good friend, Dr. Lori Thomas. We were both in college together, and she said, you probably could do more if you became a medical doctor. So her influence and her mentoring at that early stage really changed my mind about going to graduate school versus medical school. But I never lost my love for research. So at that point, when I graduated from Marquette, I actually entered into medical school after spending a year as a computer programmer, because my BS degree was in mathematics, so I love numbers. So then I entered medical school, and actually after I graduated from med school in '81, became the director of the Comprehensive Sickle Cell Program, and did very well. Was hired as an instructor. Within a couple years, was promoted to assistant professor. So I was doing great, but I had to make a critical decision. if I wanted to do research, I needed more training. So at that point, I decided to come out of a faculty position and actually go back into training to do my Hem-Onc fellowship. So it was a big decision, but when I look back at it, it was the right decision for me for where I wanted my career path to end up. And so I went into Hem-Onc fellowship in Denver, which is 3 years, 1 year clinical, 2 years of research. And again, as a subspecialist in pediatric hematology oncology, the 2 years wasn't enough. If I wanted to be an independent researcher, 2 years of bench training was not going to cut it. I was not going to be able to compete for NIH funding. So at that point, I had to decide when I finished my Hem-Onc fellowship, was I going to go into a faculty position because I was being recruited, or would I delay gratification and train more? And I was fortunate to be able to get a position in Washington, University of Washington, and spent 4 more years doing a postdoctoral training where I only learned how to do bench research so that I could be prepared to become an independent investigator. So I also was able to early on get some funding. Someone had asked me at lunchtime about funding. I was a Robert Woods Johnson Fellow, which funded my 4 years of postdoc. I had a wonderful mentor who passed recently, Dr. George Stamm, who influenced my training and reputation in the hemoglobin field. I learned true laboratory skills so that I could be confident once I was done training in the lab that I could actually have an independent research career. And during those 4 years, I finally said, okay, enough is enough training is enough. And so after the postdoc training, I went ahead and went into a faculty position so that I could support myself. But what was really good about it, because of my training, I was recruited and I was able to get the protected time I needed to build an independent research career. So when I went into my first faculty position, 75% of my time was dedicated to doing research as a medical doctor, which is I was very fortunate to be able to do that. So 94 through 2010, I went from Alabama to Texas to now in Augusta. And during this entire time, I've continued to build on my research program, 2 very important components, research, and I'm 100% dedicated to training. And I think my award from ASH was from the training effort that I've made over the years to increase the diversity of the workforce and to offer training opportunities across the board to any student who wants to spend time in my lab. So I'll talk to you about our research. That's what I'll spend the majority of the time on today. And then I'll end by showing you some of the training efforts that have gone on in the lab as well. So my research is related to sickle cell disease. Sickle cell disease is one of the most common genetic disorders worldwide. And you can see from this map, when you look at the numbers, the majority of sickle cell disease actually is in Africa. This is 75% of the world population for sickle cell disease. It's estimated about 20 million cases of sickle cell disease in the world. And the second most common place would be in India. There's a lot of sickle cell disease in India. And you hear that the United States, only 1.4% of sickle cell disease. So that translates into about 100,000 affected individuals in the United States. So you look at the numbers, about still 350,000 deaths per year from sickle cell disease. And it's mainly still due to malaria. So sickle cell disease is endemic. It occurs where malaria is an endemic. And having the sickle mutation, a trait, actually protects you against malaria. So in some areas of Africa, up to 25% of the population carry the sickle cell gene. So there's a need to not only develop treatments for people in the United States where we can do more sophisticated treatments like bone marrow transplant and gene therapy, et cetera, but to develop drugs that actually can be used for the masses of individuals affected for sickle cell disease in Africa and in India. So what is the mutation? The sickle gene, the beta globin gene, is on the chromosome 11, at the end of chromosome 11. And the basic mutation is a point mutation in DNA. Here is the normal 6 codon would be GAG, which encodes for glutamic acid. And in sickle cell disease, there's a mutation from A to T. And this single point mutation encodes for a different amino acid, valine. So the abnormalities and pathophysiology of sickle cell disease is based on this single point mutation in the DNA that leads to the production of abnormal hemoglobin S, which has abnormal behaviors in the red blood cells. So what is the basic pathophysiology? Under hypoxia conditions, hemoglobin S undergoes polymerization formation. So here is a schematic of a normal red blood cell, and hemoglobin is the main protein to transport oxygen in red cells. And normally hemoglobin is made up of 4 chains, which are in solution in the red blood cell as individual molecules. And the blood flow is maintained smoothly through the small capillaries, etc. But in sickle cell disease, because of the mutation, the hemoglobin undergoes polymerization. And the result is the production of these sickle-shaped red blood cells that cause vasal occlusion in the small capillaries because these cells are very stiff and not pliable. And the net result of that, of course, is tissue ischemia downstream of where the vasal occlusion, and the most common clinical symptom that we observe would be repeated episodes of pain. But it can also affect other parts of the body. It's a form of stroke, cardiac disease, the spleen can be affected, etc. So what has been the treatments? The mutation for sickle cell disease is we've known it for over 100 years, what's caused the mutation and so forth. And it was the first really genetic disease that was defined at the molecular level. So it took many years before we got our first FDA-approved drug, which is hydroxyurea, was approved in 1998 after a big multi-center study. And then it took from 1998 to 2017 to get it approved for children. So when we were using it in children up to 2017, we were using it as what we call off-label, just based on the work in adults. Hydroxyurea is a ribonucleotide reductase inhibitor, but its main benefit in sickle cell, it donates nitric oxide, which helps with the blood flow and function of the vessels, and it induces fetal hemoglobin, which is my main area of research. And now is considered the standard of care for sickle cell disease. So fast forward, it took another 20 years to get our second drug. So just recently, Indari was approved in 2018, although many drugs were being tested. So finally, Indari was approved for children and adults, and basically it helps to prevent the oxidative damage of the red cell membrane from sickle cell polymerization. And Indari is actually a very benign compound, is L-glutamine. And it becomes a precursor for the production of glutathione, which is depleted in sickle red blood cells. So we're just starting with Indari. In the study, they showed that you could combine Indari with hydroxyurea and get even a more beneficial effect. So it's sparking a lot of discussion about combination therapy, where you have 2 drugs that are acting by different mechanisms, which is common in other treatments like oncology, et cetera. So this is where the field is moving now, to develop combination therapy. So hydroxyurea, which is the standard of care, has a lot of benefits in sickle cell disease. The biggest one that they showed in the study was that it induces fetal hemoglobin, and I'll tell you why that's important, okay, in the bone marrow, in the erythroid progenitors. It also decreases the neutrophil count, which there's a lot of inflammation in sickle cell disease, so it helps to quiet down the inflammation. It decreases the adhesion of the sickle cell with the blood vessel. It reduces the breakdown of the red blood cells, so the hemoglobin level actually improves, and it acts as a nitric oxide donor, which helps to improve again, the vasculature and decrease in damage. So what's the evidence for fetal hemoglobin, which has been a big driver in the field? This was from the natural history study of sickle cell disease that was conducted for 20 years. And what they showed following 4,000 patients for 20 years is that the long-term survival in sickle cell disease is directly correlated with fetal hemoglobin levels. So individuals with greater than 8.6% fetal hemoglobin had improved survival. So normal fetal hemoglobin level is probably like less than 1%. So you can see that it doesn't take a big increase, but just having an 8.6% level. Here are the patients with low fetal hemoglobin. You can see the probability of survival is much lower than the individuals with high fetal hemoglobin. So this became the driving force for a lot in our field for developing fetal hemoglobin-inducing drugs to treat sickle cell disease. Why does fetal hemoglobin work? So this is a schematic showing you in untreated patients, if you look at the blood flow, under hypoxia conditions in the small vessels, you get vaso-occlusion and ischemia. But in someone who's been on hydroxyurea, if you can achieve 25-75, so 25% fetal hemoglobin, 75% sickle, what you see then under hypoxia is that you only get small amount of polymers or no polymers. And the net result is you can maintain blood flow to the tissues. So this is it. Fetal hemoglobin actually directly inhibits sickling of the sickle hemoglobin, so that's why it's so effective. And in fact, people have genetic mutations where they have what we call hereditary persistence of fetal hemoglobin, and if they have that along with the sickle cell disease, those patients are asymptomatic. They don't have any of the symptoms like regular sickle cell anemia. So we know from nature experiments and from work in the labs that inducing fetal hemoglobin is very effective. So I always have to show this slide, and most people are like, oh my God, the switching slide. But this is fundamental to understanding sickle cell disease and hemoglobin switching, which is what the whole field is about in studying. So on chromosome 11 are the beta globin-like genes. There's this region called the locus control region, which is a big enhancer element that actually throughout development interact through looping with the individual globin promoters to actually accomplish gene expression at different stages of development. So this graph right here is showing the different beta-like globin genes. You have the embryonic, the 2 gamma genes, which produce fetal hemoglobin, and then you have the beta globin gene, which produces the adult hemoglobin. And this is where the mutation is in sickle cell disease. So from conception to about 6 weeks, in the yolk sac, epsilon hemoglobin is produced. And from 6 weeks up until after birth, high levels of fetal hemoglobin are produced. And then after birth, the fetal globin genes are turned off and the beta globin genes are activated. So in general, we don't see symptoms from sickle cell disease until around 6 months, where the level of the sickle hemoglobin has gotten high enough to cause the vaso-occlusion and start to produce some of the clinical manifestations. So a key to this whole system is, how can we reverse this process? Better yet, how can we prevent the gamma to beta globin switch to, in effect, make patients like the genetic mutation for hereditary persistence of fetal hemoglobin? And you can see from the natural history study, you don't need to get really, really high levels. Just enough fetal globin and all the red cells will be effective. A lot of when the GWAS, genome-wide association studies came out, genetic studies, we had a big breakthrough in the field when the BCL11A protein was discovered. This is a transcription factor that is tightly linked to fetal hemoglobin, inherited fetal hemoglobin levels. And what this whole system showed is that BCL11A, which is a repressor of gamma globin, is actually activated by a second transcription factor called KLF1. And this is a master regulator of helping to silence gamma and to activate beta. So BCL11A has been studied extensively, shown to interact with multiple transcription factors, and these now have become targets for gene therapy and new drug development, drugs that can inhibit BCL11A, etc. And also there was an enhancer element identified in the BCL11A gene that is what is being used to actually develop gene therapy for sickle cell disease. So this molecule is critical. A lot of talk about, can you target a transcription factor for treatment, because they're very difficult to target. But The group at Harvard has been able to master it. So we're very encouraged that there will be, BCL11A is what we call druggable. Drugs can be developed that will modulate the expression. Now, one caveat to that is BCL11A is important for B cell development, and it's involved in some neoplasm. So there was concern that if you manipulate BCL11A with individuals end up with the cancer. But using this enhancer element is kind of alleviate that concern because you're not manipulating the entire gene, just that enhancer element. So what is an ideal drug? A lot of things make an ideal drug. First of all, it works. There are a lot of drugs on the market that don't work, even though they're on the market and FDA approved. So high efficacy, it works for its intended purpose. Cost needs to be cheap. Even in the US, we had difficulty getting hydroxyurea for our patients in South Georgia. And so you can imagine what it'd be like in India and in Africa, where that's where the masses of sickle cell patients are. So you want something that's cheap, something that's easy to administer, and of course oral route would be the best, and stable. We were talking about going to get refills, you know, on your medication. In young babies who we treat with penicillin to protect against infection, those parents have to go every 2 weeks to pick up the penicillin. Do you imagine? Every 2 weeks, because it's a liquid, and it's only stable for 2 weeks. So if you're talking about a lifelong treatment, you have to have something that is stable and will require the least amount of going to pick up refills. Predictable mechanism of action. You know, under different circumstances, will the drug act different? So that's important. Reversible action. So, for instance, hydroxyurea. It's a chemotherapy drug. It causes bone marrow suppression. That's why we have to monitor it, but it's reversible. So that if it's a very specific dosing, and we start at a low dose and titrate it. So if there's too much suppression, we can stop it and reverse the effects, and then we just start back at a lower dose. So the drug being reversible is very important versus a drug that would cause irreversible toxicity, and you wouldn't have any fallback to, you don't want to do any harm to the patient. Low adverse effects, you know, the more side effects a drug have, the less likely, you know, any of us are going to take it long term if you're having side effects every time you take your medication. So low adverse effects and side effects, and then interaction with other drugs. You know, a drug where you can only take it by itself and you can't take it, you know, the grapefruit, it diminishes the absorption, and then certain drugs you can't take together. So the more complicated the interactions, the harder it is for someone to take a drug long term. So this is a lot to live up to for an ideal drug, but no drug is ideal. But some of these things on the list are critical, you know, for me, like this right here, toxicity. And with all my clinical work, adherence is top of the list, because even though hydroxyurea is effective, if the patient doesn't take it, it's not effective. So adhering to the regimen and having something that the families can get easily is critical from a medical physician perspective. So there's been a lot of effort to develop drugs, and this is the short list. This doesn't really account for all the different drugs, but I just highlighted the critical ones. Of course, hydroxyurea and Endari, which I've already commented on. And in the late 90s, arginine butyrate, which is a HDAC, histone deacetylase inhibitor, was shown to be very effective, more potent than hydroxyurea. But guess what? Arginine got left behind because the route of administration, you can only give it IV. So even though it was a better drug, it had to be dropped clinically because there's no one who's coming, you know, 3 times a week to get an IV dose of medication. So the route of administration killed arginine butyrate, but it led to a lot of other effort to develop HDAC inhibitors that you could take by mouth. Other drugs, this new drug, one of the MABs, crizanlizumab, there's thousands of these MAB drugs, was just finished, a monoclonal antibody against P-selectin. And this is a protein that's expressed in the endothelium and that causes vaso-occlusion, helps to initiate vaso-occlusion. So this monoclonal antibody actually blocks that interaction. And that study was just completed in 2017, showing that you could add this drug to hydroxyurea, and it improved, decreased the rate of pain episodes. This drug still needs to go through phase 3 trial, so it's still under development. It's not FDA approved, but very encouraging that we'll have a monoclonal antibody for treating sickle cell disease. And then there's some other derivatives. This drug, pomalidomide, was not effective as a fetal globin inducer, so that was completed. Recently, decitabine, which is also a very potent fetal hemoglobin inducer, but the problem with decitabine by itself was once you take the drug, it was inactivated immediately. If you give it IV, it worked. Give it by mouth, inactivated. So they decided not to stop with decitabine. What they did was made a formulation to stabilize it with this tetrahydrouridine. So it blocks the enzyme that metabolizes decitabine. And they just finished a trial in 2018 to show that was effective. So now that drug is under development, hopefully for FDA approval, so that we'll have a second fetal hemoglobin inducer that we use in sickle cell disease. And then there's some other HDAC inhibitors. This one's in phase 1. And today I'll talk a little bit about the effort in my lab to develop Benzeraside and the dimethyl fumarate, both as fetal hemoglobin inducers. So one of the articles that I sent for your seminar discussion was related to, I chose 2 articles that were a little bit different. One was about high-throughput screening and how you can go through drug development. And I've done this in collaboration with Susan Perrine, who's my lifelong friend, and we've been working together in like the last 20 years. That's the other thing. If you want to develop drugs, you have to be in it for the long haul. There's nothing quick in drug development. And so I sent this one related to our system for high-throughput drug screen. Then I sent the second paper related to dimethyl fumarate. So let me just briefly, I was told you didn't do this paper. So let me just briefly go over the findings we have for the high-throughput screen. And the goal was to establish a high-throughput screening system to identify novel fetal globin inducers, which could be developed for treatment of sickle cell disease. What we had was a reporter construct that was made of green fluorescent protein, the gamma promoter, the gamma gene promoter, and then part of that locus control region. This small construct was transfected into K562 cells, and then we screened a 10,000 FDA approved. We decided to use FDA approved drugs. We screened these drugs for the level of fluorescence that was produced with each of the different drugs screened. And this is just showing you an example of control with no drug, and this was the arginine butyrate that I told you was a potent fetal globin inducer. And you can see GFP is expressed in almost all the cells. So it's a wonderful positive control. So we screened 10,000 compounds, and out of that 10,000, these are the ones that had increase in GFP that we thought were important enough or enough change that we wanted to pursue further. So you can see out of 10,000 compounds, we didn't get many hits, but we did get some strong hits. The GFP increased significantly for this one. The label's not here, but this compound over here on the end, and I'll have more on the next one. That was the idarubicin. But drugs like idarubicin are cardiotoxic, so we decided not to go forward with something like idarubicin. It uses chemotherapy. It can damage the heart muscle. So we wanted a balance between GFP induction, but also a drug that was not toxic. Because when you're doing treatment for sickle cell disease, it's different than treating someone with cancer. Sickle cell disease is not a malignant disease, so your drug treatment can't be as strong in general for sickle cell disease like you would someone with cancer, where, you know, if you don't do something, they may die, you know, immediately. Sickle cell disease, we can't predict, you know, how bad the disease is going to be, and they may live another 40, 50, 60 years. You know, the average lifespan now is in the mid-40s. So you have to use a different approach when you're trying to develop a drug for sickle cell disease than if you were treating cancer. So there's basic techniques that we use in the lab, which go across a lot of different specialties, and so I just wanted to take a couple seconds to talk about these techniques. You know, you will learn about these when you read papers in the literature. You need to understand what these techniques involve. So the main way we measure gene expression is through reverse transcriptase quantitative PCR. Fairly, you know, what seems to be simple now took many, many years to develop, and it revolutionized our ability to look at gene transcription. So in general, you isolate your RNA from whatever cells that you're working with. You do a reverse transcription reaction to produce the complementary DNA. And the difference between regular DNA and complementary DNA, anybody know the difference? Yes, ma'am. Wonderful. The introns have been spliced out. So basically, you're reverse engineering the complementary. So wonderful. There's a good teacher. And I was like, okay. So then you take this complementary cDNA and then run a forward quantitative PCR reaction. So the RT is a reverse reaction, and then the qPCR is a forward. You have your cDNA. You got to have primers for your gene that you're going to quantify, and you have a fluorescent probe. And these days, almost everybody uses CyberGreen as their fluorescent probe. So you do a simple PCR reaction, but the difference is you have this fluorescent probe. You denature your primers anneal, and then Taq polymerase, along with nucleotide DNTPs, make copies of the DNA, and during that time, the CyberGreen intercalates in the DNA. And you have a fancy machine. This is the one we have in the lab, Biolab, but there's other ones, ABI, et cetera. And this machine actually quantifies the level of CyberGreen based on how much starting cDNA was there. So it gives you a quantitative measure of your gene expression. So you've gone from messenger RNA, a total RNA, to cDNA, and it's only identifying message, and then you're quantifying the gene expression. So we did that for these high potential agents, and what we saw when we looked in K562 cells, so now this is not GFP, this is the actual gamma gene itself in K562 cells, that the findings were pretty reproducible from the GFP. And so the drug, here's arginine butyrate here, and Benzeraside is what we became very interested in because, again, it's a drug that's FDA approved, and it has a strong induction of gamma-globin, almost fourfold. So it would be predicted to be enough to have a clinical effect. These other agents we didn't pursue because they have more toxicity based on their structure. So we confirmed the gamma gene expression using RT-qPCR, and then we also did immunoblotting to show that actually not only was the gene increased, we also saw an increase in actual fetal hemoglobin protein. So it's not enough to just say the gene increased, because the clinical benefit is from the protein, not from having more messenger RNA. So that mRNA has to be translated into more protein. And Benzeraside, as I said, it's been used for decades, 4 decades in the UK and Canada. It's not FDA approved in the US, but it has been used and safely for Parkinson's disease. So it's nice to have a drug that's already been tested in humans and established as effective. So then we wanted to see whether or not Benzeraside would induce fetal hemoglobin. Actually, we used a baboon, a large animal model. So the baboon is actually made anemic to mimic sickle cell disease, and then they're treated with the drug. You can see up here the different treatments shown in black. And we saw induction of gamma globin by about day 12 or so in the baboon, and we were very, very pleased with the fact that the hemoglobin level improved as well. So for sickle cell disease with the severe anemia, you get the fetal induction and the improvement in total hemoglobin level, which would be very beneficial. Then we wanted to say, okay, if gamma globin's increased, do we see more protein in the blood cells? So we do an assay known as flow cytometry, which is, again, another very commonly used assay that is, you get a lot of bang for your buck, you get pretty pictures, and you get a lot of good data. And I won't say it's easy to do, but it's not as challenging as some of the other assays we do. So basically, the baboons that have been treated with the Benzeraside, we draw blood. I shouldn't say we, the veterinarians draw blood. We don't work with the baboons directly. So in the animal facility, they draw the blood and then give it to us. And then it's stained with a fluorescent antibody, FITC, and then flow cytometry is done. And how many are familiar with flow cytometry? Okay, good, good. So basically, you have your preparation of cells that you want to analyze, and then the sheath fluid goes into the machine. It's a big, fancy, expensive machine, but it's great. The cells are then funneled through for the laser detection in single cell order, and as the cells pass the laser, if a cell is positive, in this case they're using red fluorescence as an example, but for us it would be green fluorescence, then the fluorescence, the laser activates and the fluorescence is emitted, so you can measure how many fluorescence, and then you also measure a forward and side scatter that gives you a cell count. So in the end you can say what % of the cells actually had your protein that you're assaying for, and in our case it would be fetal hemoglobin. And it's showing in the baboons, not only did we see gamma globin activation, but fetal hemoglobin increased significantly. These are the number of red blood cells that had the fetal hemoglobin expression, so they're called F cells. And we also saw an increase in what we call F-retic, so these are young red cells. The orange shows the reticulum stain with acridine orange, and then the green shows the fetal hemoglobin in the cytoplasm. So these are reticulocytes, which are early red cells, that high level of fetal, and these are just the fetal stains. So we were able to show in the baboon that Benzeraside increased fetal hemoglobin in both the mature and the reticulocytes. So how do we, for the long stay, when you look at drug development, we've been working, Benzeraside actually was a shorter amount of time. We've only been working on that drug for about 7 years. But this is a typical timeline for drug development. If you're going from discovery, some of our early agents, I was sharing with the students at lunch, some of our early agents that went from bench to bedside, they didn't work. So we had invested 15 years, but that's why we changed our strategy to go with FDA-approved drugs. So that once you've done the research and the preclinical in mice or baboon, then you file for an investigational new drug license from FDA. Then you get into the clinical phase, phase 1, small, phase 2, phase 3. And if it's working, then you get a new drug approved, here filed, and then you finally get your drug approved. So the average time to market is about 15 years, and probably even longer, to be honest. And then you get 5 years on your drug patent. So you take 15 years to show that it works, and then you get 5 years on the patent. So it's a challenging field, but here are the numbers that they quote in this example. You start with 10,000 drugs, there might be 20 that make it to market. But it's needed, because we need more treatment for individuals with sickle cell disease. And then the second agent that we decided to work with is dimethyl fumarate. We got very interested in this drug because it was actually approved for the treatment of multiple sclerosis. It's a small molecule. It had oral formulation, so it met a lot of our criteria for a good drug to be developed. And this drug, dimethyl fumarate, is actually hydrolyzed in the gut to the active compound monomethyl fumarate. And it also has neuroprotective effects, anti-inflammatory effects, through the activation of this key protein, NRF2. So in 2013, when Tecfidera was approved for the treatment of multiple sclerosis, we went into partnership with Biogen to look to see whether or not it could be used for sickle cell disease, because we knew NRF2 was important for globin gene regulation. The Tecfidera is actually BID dosing, which is not ideal. Hydroxyurea is just once a day. So you have to double the amount of times you take the drug. Guess what? The adherence is going to go down if you have to take it twice a day. So this is a little bit of a drawback for DMF. But it does not suppress the bone marrow like hydroxyurea, and it's not a chemotherapy drug. Hydroxyurea is chemotherapy. So patients are sometimes reluctant to use a chemotherapy drug. So to test DMF, we started in what we call a primary culture system. These are very useful systems for looking at the ability of drugs to induce the gamma-globin genes. We start with CD34 cells from sickle cell patients, grow them in culture for about 7 days, and then withdraw these two factors to get erythroid differentiation. And you can see here, we go from early progenitors all the way down. By day 10, we see the hemoglobin in the cytoplasm of the cells. And by day 14, the cells have actually enucleated, so we get mature cells. And you kind of, this curve reminds you of the switching curve. So in our erythroid progenitors, we actually can observe hemoglobin switching, just like we do during development. So during erythropoiesis, the gamma to beta switch occurs, and that allows you to hopefully get some insight on mechanisms for the drug treatment and how we can reactivate gamma. So we treat with this drugs on day 8 for a couple of days, and then we do our analysis similar to what I talked about in the baboon. So for sickle progenitors, dimethyl fumarate caused a very nice increase in F cells. Here's a 200 micromolar DMF compared to hydroxyurea, which performed a little better than the DMF. And then we did combination to show that if you use a different mechanism, you can actually get an additive effect for fetal hemoglobin induction. And then here's the actual protein by Western blot. Again, we see that adding the two drugs together, you get a nice induction of fetal hemoglobin. And DMF produced an anti-sickling effect. We talked about the polymerization of hemoglobin S. So under hypoxia conditions, we see an increase in sickle cell formation. This is 3 different patients under hypoxia, but if we pre-treat it with DMF, we're actually able to block the sickling. So in 2 out of 3 patients, there was an actual physiologic effect to block sickling. 1 patient did not respond, which is not unusual. Even with hydroxyurea, we don't get a 100% response, so not all patients are going to respond. So this was very encouraging that it actually had an effect that could be important clinically if we block sickling. So then we wanted to turn to, and this I believe is the second paper that I sent, and try to understand mechanisms. How is DMF inducing gamma globin? Because not enough when you want to publish in high impact journals to just say it induces, you have to tell the reviewers why, what is the mechanism. So we looked at NRF2, which is a potent transcription factor that controls oxidative stress in cells. So when a cell is under low stress, NRF2 is actually sequestered in the cytoplasm and degraded in the proteasome because there's no need to have high NRF2 levels. But we speculated that when we treat with DMF, that DMF actually could block KEAP1 and allow the release of NRF2. And then NRF2 then could translocate to the nucleus and bind in the promoter of either its target antioxidant genes or to the gamma globin gene. So this was our working hypothesis that we tested. So we can see after DMF treatment, we saw activation of gamma globin in these primary cells, and we saw activation of the heme oxygenase 1 and NQO1, which are the oxidative stress genes at the mRNA level and at the protein level. Here's NRF2, fetal globin, and what was neat was we did not see any change in sickle hemoglobin, so the drugs, some drugs like hydroxyurea actually increase the beta globin expression a little bit as well. So it's that balance between the two, so we're encouraged by that. And then here are the antioxidant genes. Then we did shRNA knockdown of NRF2 to see what effect they would have on gamma globin expression. We transfected primary cells with the shRNA molecule, and we saw there was a drop in not only gamma, but the other epsilon globin gene as well, which is considered a very early expression, but no effect on the beta that suggests that NRF2 is needed for gamma. And then we looked at F cells by flow cytometry. Again, a drop in F cells, not as dramatic as when we treated with the drug DMF, but definitely a significant decrease. So the next question we asked is, what does DMF do to the nuclear, the location of NRF2 once the primary cells have been treated? So we did separation of cytoplasmic and nuclear protein, and then we did Western blotting to look at the level of NRF2 with and without DMF. And we can see that the change really is in the nucleus. There's a much significant increase in NRF2 in the nucleus after DMF. And this is just immunohistochemistry showing the DAPI to stain the nucleus. And then in untreated cells, NRF2 is mainly in the cytoplasm. So that's the red dots, that's NRF2 in the cytoplasm. But then when you treat with DMF, here's DAPI for the nucleus, the staining for NRF2, and when we do the overlay, we can see that NRF2 is mainly in the nucleus now. So this showed us that DMF actually promoted translocation of NRF2 into the nucleus, where it might then bind to the gamma-globin promoter. So the last assay we did is called CHIP assay, chromatin immunoprecipitation, and it's a powerful tool that most labs are doing routinely. And what it does, it evaluates in vivo, in the live cell, DNA-protein interaction. It's one thing to demonstrate it if you have an in vitro system where you're just adding DNA to protein together. But this is actually in the primary cell. Does NRF2 bind to the gamma promoter? So basically, you treat your cells with whatever drug you want to test. You do the formalin, you lyse the cells, formalin cross-link, lyse the cells, and then you want to, now you have your DNA protein complexes established. You sonicate or fragment the DNA, and then from there you do what we call an immunoprecipitation reaction. So you pull down the protein that you're interested in quantifying. And once this is accomplished, you can purify the DNA and then actually measure the level of that chromatin-protein interaction using the CHIP assay. So we were able to show, using CHIP assay, that in the gamma-globin promoter, this NRF2 binding site that we identified at -100, that there was increased binding of NRF2 after DMF treatment. No change in tidal binding protein, which is a general transcription factor in activating gene expression. And then we also looked in that locus control region, hypersensitive site 2, showed there was a big increase in NRF2 binding after DMF in HS2 as well. And then we looked at a negative control region to show that there wasn't really much change in a region where there was no NRF2 binding. So it's just important to do negative controls as it is to do positive controls. So with that, we really felt like NRF2 was critically involved in the mechanism of gamma-globin activation by DMF. But then we also wanted to expand, is NRF2 important for just gamma-globin expression in general when you're under non-drug treatment conditions? So we started working with our sickle cell mouse model, showing here that actually this color with the change. And Tim Towns developed this model in his lab in 2006. And what it is all the mouse genes are knocked out. And these mice only express the human hemoglobin genes. In addition to the alpha that make true hemoglobin, they have the A gamma, normal beta on a small construct. And then they have a second construct with the normal gamma and the sickle cell gene. So this mouse model allows us to make all genotypes, SS, sickle cell trait, and AA, but it's all human hemoglobin produced. This animal has sickle cell disease. It has markedly enlarged spleen from the chronic hemolysis. Here's a normal, this is AA, and this is the SS mouse. Sickle cells, they have pain. A lot of research is done with pain in the sickle cell mouse model, and they also have decreased survival, so they're a very good representation of human sickle cell disease. So we treated the animals with DMF for 4 weeks, along with hydroxyurea, positive controls, and then the vehicle. Both of these drugs were dissolved in water. So we treated Monday through Friday, 2 days off for 4 weeks. And then at week 0, 2, and 4, we collected blood samples for our analysis to see what happened with the fetal hemoglobin level. And then we also did a sickling assay to see if they were sickling. And even in the sickle cell mouse, we saw a significant increase in fetal hemoglobin in vivo, about a 3-fold increase in fetal hemoglobin levels. And we were able to improve the hemoglobin. The average was 7. The normal for the mouse is about 12. But the sickle cell anemia of 7 is about average, and then we were able to increase it to almost 9, so almost 2 grams of hemoglobin improvement, which would be critical for a sickle cell patient to have a better hemoglobin level. And these are the hypoxia studies showing that in the sickle cell mouse, that basically you see a decrease in sickling with DMF, and in 2 of the mice, and then in the 3rd one, there was no effect. So then the last experiment we did was to knock out NRF2 to see if it was important for just regulating gamma-globin independent of drug treatment. So this is a common approach. We took the sickle cell mouse and we crossed it with the NRF2 knockout mouse, which was actually available with one other investigator on our campus. He gave us these mice. And after about 2 years of breeding, very complex genetics, oh my God, when the junior faculty asked me to do that, I had no idea what he was asking me. And then once we got into it, I was like, oh my God, help me. So once we were so far into it, we couldn't turn around. So we kept going until we finally got it. So this is the B6 wild type showing the proper fragment. This is PCR. And here is the NRF2 wild type mouse here. And then here is the heterozygote showing the loss of the NRF2 band. And then the complete knockout showing that there was no NRF2 production. So we were pretty sure that we had our knockout. And we were very pleased to show that he was able to harvest the spleens from these mice during development and show that when you knock out NRF2, the level of gamma expression is decreased by almost 50%. And there's hardly any detectable fetal hemoglobin when you knock out this gene. So it substantiated its role in normal hemoglobin regulation, not just under drug treatment conditions. And this is the peripheral smear from the mice showing under normal conditions, normal oxygen conditions, the wild type and knockout. And then under hypoxia, there were more sickle cells, which became significantly increased in the NRF2 knockout mouse. So showing that NRF2 is important for gene expression, globin gene expression during mouse development. And this work was published in Blood recently. So DMF, just to summarize, we're still very interested in developing DMF. And we think that it works by inhibiting KEAP1 to release NRF2, which then can translocate to the nucleus and activate gene expression. In particular for us, gamma-globin. I didn't have time to go through the other studies we did. This paper was published in JCI in 2017, but we also looked at its anti-inflammatory effects, because it's known to be anti-inflammatory in multiple sclerosis. So it decreased the level of inflammation and liver inflammation. It decreased the level of plasma-free heme, which is detrimental in sickle cell disease. So we feel that DMF not only will induce fetal hemoglobin, but will have other positive effects similar to hydroxyurea that makes it an attractive drug for developing. So when you have high fetal hemoglobin, again, you hopefully will block the sickling in the red blood cell, reduce sickling, reduce hemolysis, and diminish inflammation. So hopefully it hits multiple targets in individuals with sickle cell disease. And we sharing with the students, we were just at the point where we finished the preclinical studies, and we wrote the protocol for the phase 1 clinical trial. But the company then split, and we didn't have the money to do the phase 1 trial. So now we're at the stage where we need to find funding to do the phase 1 clinical trial to see if DMF will be useful for sickle cell patients. So we haven't given up yet. So there's all kind of factors that can come in to just blow a hole in your plan. So that's what happened with the DMF, but we're still working on it. So the last thing I want to say in the last couple minutes of my talk is about my training. I've talked to you about my research. And the other big part of my career path as a physician scientist is training. I felt very, very compelled and dedicated to training the next generation of scientists. It's so important. Mentoring is critical. Of making available in my lab opportunities to do research. And I've trained young investigators in junior high, high school, all the way up to junior faculty level. So I've trained a lot of people over the last 30 or so years. And I just put this one slide together to summarize the number of people who have gone through my lab that I personally trained, over 80 individuals. I'm really proud of the fact that 60% were underrepresented, so that I can help to diversify the workforce. 60% female, so it's not that I'm favored, but women tend to gravitate to my lab. High school kids, a lot of undergraduates, students doing thesis projects, etc. Medical students. I've had a medical student in my lab almost every year doing summer research. Master's thesis as a major advisor. PhD thesis for 10 students who've gotten their PhD through my lab. Postdocs. And then most proud of the junior faculty mentoring program that's funded by NIH, where we actually train underrepresented junior faculty from around the country. We've been doing that since 2006, and we've trained 76 junior faculty to conduct research and to try to improve the biomedical workforce diversity. So that's why I got the ASH Award, is for that NHLBI training program, and of course for the other students who have gone through my lab. So I'd like to just acknowledge the people that I've worked with over the years. Susan Perrine has been really a critical collaborator for the drug development. We continue to work together. My lab members, Dr. Zhu did most of the work you saw for the Hematologica and the blood paper. Dr. Lee, who's very strong, he does all of my primary culture work, couldn't get anything accomplished without him. And then, of course, funding from NIH for my R01 and R25 training grant. Thank you.
Thank you so much, Dr. Pace. We have time for a few questions or comments.
So I know that you have said that you're in a position now where you want to work in a sphere of having drugs that have already been FDA approved, but sort of stepping back from that to the KEAP1 NRF2 sort of mechanism. Do you have ideas as to why and how DMF is sort of preventing KEAP1, not only sequestering it to the cytoplasm, but also KEAP1 acts as an E3 ubiquitin ligase? So maybe not for your research, but for other people doing drugability studies, how to, in a sense, block that kind of.
So DMF actually directly inactivates KEAP1 by breaking down a disulfide bond. So that's how it inactivates KEAP1. So it's very specific for that molecule, which then releases, it's really not stimulating NRF2. It's what NRF2 is present in the cytoplasm is now free to translocate to the nucleus. But you're right, there are other drugs that may target that same mechanism, and there really are. We just haven't done research with them, but you could use any drug that would inactivate KEAP1, and in fact, they've done KEAP1 knockout mice and shown that it makes a difference in fetal hemoglobin. So the mechanism for DMF, it has been established. It breaks down the disulfide, the KEAP1 is inactivated, and it releases the NRF2. There are other drugs that can activate NRF2 at the gene level. We haven't tested any of those, but that would be another possibility. But given the length of time it takes to develop a drug, that's why we decided to go the route of drugs that were FDA approved and proven to be safe in people with multiple sclerosis.
Is it a question? Okay. I'm intrigued by the use of DMF and MS because when you think about the CNS effects that occur in sickle cell disease, not all of them are caused by the inclusion. There are interactions that we're not quite aware of exactly what happens. So it's intriguing to me that we might be able to find a drug that does something other to
absolutely. That's kind of the multiple targets that one drug might be able to hit. Like, we were all surprised when hydroxyurea had anti-inflammatory. Nitric oxide, well, that wasn't that surprising because of the molecule of hydroxyurea, it actually donates to nitric oxide, but that was beneficial. Absolutely, for DMF, we were very interested, could it have some neuroprotective effect in the central nervous system, because 10% of children with sickle cell disease have strokes. And we know that the vasculature is abnormal, the interactions, the inflammation. So since DMF can treat multiple sclerosis, our hope is that we would see some positive effect, absolutely. So that was just one more reason why we really try to go after the DMF, and it's anti-inflammatory. I didn't have the chance to show the data. We show a clear decrease in inflammatory cytokines in the plasma, irritation and inflammation in the liver. So we're hoping that it would have multiple effects and definitely some neuroprotective effects.
So you're probably looking for about $2 to $3 million then to first phase 1?
Yeah.
If I could just follow on that for a quick second. So early on, you reminded us that hydroxyurea leads to production of nitric oxide as a donor, but also activates soluble cyclic GMP, I guess, through one of those cyclase. So my first question is just what about the dual action? You get the donor. If you activate one of the cyclase, you get more NO as well. How important is that? Why don't you just use a donor, L-arginine, to feed the NOS pathway?
Actually, L-arginine has been tested, and it was not a good fetal globin inducer. And it actually caused more sickle symptoms. So it was discontinued in a clinical trial. So yeah, you're thinking that, and the people who did the trial thought that way as well, but it ended up not working, L-arginine. The cyclic GMP activation, we think that actually is part of the mechanism for fetal hemoglobin induction by hydroxyurea. Cyclic GMP, then actually that whole pathway is activated in gamma globin. They've shown very clearly that it's involved in gamma globin activation. But that balance, the cyclic GMP, that would increase the nitric oxide. So we have it from two sources, the cyclic GMP. And people actually right now, I think in clinical trial, there's a drug that activates cyclic GMP for testing in sickle cell disease. And the word is not, we don't know what the outcome is yet. But they're looking at it for the nitric oxide doning potential, and they're keeping quiet on whether or not it induces fetal hemoglobin as well. But definitely it's a strategy. Some companies have decided to just target the cyclic GMP.
I also just quickly ask you, so is sickle cell disease, can it be thought of as also the disease of the vasculature? And downstream, when the damage is done, the inflammation, even if you repair the sickling, you still have the effects on the vessels. And so is that part of it?
So now he's getting into the controversy in the field, and there is a lot of evidence that it's a basic genetic mutation that produces the sickle hemoglobin. So as a purist, as a fetal hemoglobin person, I say, if you correct the basic problem early on, then you don't get all the downstream, we call them downstream effect, but you have the people who are the downstream effect people. So they say we should develop drugs, because it is true, even though you may induce fetal hemoglobin, you may not reverse the downstream effects. So that's why the field is moving toward combination therapy. So you'd have a drug, hopefully that would be safe to give to young babies before the switch is completed. And the goal would be, as a pediatrician, it's like, I need something that's going to prevent the damage, because once it's done, it's hard to reverse it. That was shown in the baby hug trial. The spleen damage, once it was done, even though the fetal hemoglobin was induced, the spleen was still damaged beyond repair. So yes, we want drugs that can be safe, started early, but there is still a very, very much need to have drugs that can treat the downstream effects. Yeah.
Anybody else reading the middle? Your question?
I've got a question. I just want to make sure that I understand the slide correctly. This is the mouse study with the sickle cell mouse model where you got a vehicle treatment, hydroxyurea treatment, and the DMF treatment. How did DMF compare to the hydroxyurea treated mice? And did you think about a combination therapy in that study?
That's a very good question. Hydroxyurea still performed better than the DMF in the sickle cell mice. We did combination therapy in sickle cell progenitors, but we didn't do it in the sickle mouse model, because the sickle mice are pretty fragile. So when we did those studies, we elected not to do the combination, but absolutely that would be needed, I think, to give more evidence for getting that IND, that you have an additive effects when you add the two. Our tissue culture suggests that will be true, but you got to weigh that against the increased toxicity when you combine hydroxyurea with DMF. It's great that DMF doesn't have any bone marrow toxicity, but the mice may not be able to tolerate it. So that was the only reason why we didn't do the studies up front, but it's needed.
Throw a hand up there.
Hi, you mentioned previously that sickle cell has a sort of protection effect against malaria. And I was just curious, are HbF positive cells susceptible to the effects of malaria?
That's a very good question. So having a sickle mutation protects against malaria, and it's believed that it keeps the malarial parasites from having the oxygen that's needed in order for it to propagate. So I would predict that it's going to be a balance between the beneficial effect of fetal hemoglobin versus having the sickle hemoglobin. And we wouldn't treat someone with sickle trait with to induce fetal hemoglobin anyhow, but you're asking a question about when you combine malaria with sickle cell disease, which is worse. And in general, if you have sickle cell anemia and you get malaria, the disease is much worse because of the underlying chronic sickle cell anemia. So I would think overall, having a higher fetal would still give you a better outcome if you had sickle cell anemia and contracted malaria, because it would block the polymerization. But you know, how much would be enough? How much would be a benefit versus, having sickle cell anemia and malaria? But those children do much worse than a child without sickle cell anemia.
Please, okay, one last question.
So this doesn't have entirely everything to do with science, but I think it's really important we're able to, like after this point in time, be able to create such diverse culture, like through your lab. So in what ways, was that something that you started off, you're like, I want to be able to create something like this and have 60% underrepresented folks or 60% people in this lab? Or was it something that like after the fact you look back on and you're like, oh, that was really interesting?
You mean creating the culture system itself, or?
Yeah.
Well, it was back when I first started working in the lab, believe it or not, these primary culture systems were not established. It was very difficult. We used cell lines for years, but they were kind of rejected because, yeah. Okay.
But like,
Like the culture of your lab.
The students and the trainers and the diversity.
Okay, what about them now? Oh, you mean that I have a goal to have a diverse lab? Now I got it. Well, when you said culture, I was thinking like tissue culture. Yeah, culture the students too. Okay. I set out to offer training experience to anybody who wanted to have a training experience in my lab. And this is just, I think, because of my diversity and inclusion attitude, I attracted a wide range of students to my lab. But then also I tended to attract more underrepresented students as well because of the culture in my lab. So I would say my numbers of 60% underrepresented is way ahead of the average lab. The majority of basic research labs might have 10%, one or two students every so often, but they tended to gravitate to me, and the same thing with the females. So I didn't start off with that goal, but because I wanted to help all the students who wanted a research experience, because it's important to help anyone who comes knocking at your door. That's the way it turned out. So it was a culture that was cultivated.
Please join me in thanking Dr.
Thank you, sir. Thank you. Thank you. Thank you. Thank you. I know.